Good morning, all. This is Ram Santhanam. I'm the Global Account Manager for the Targeting Sequencing business. Glad you could all join. We're extremely excited about the topic of targeted sequencing, as there's been tremendous interest, right? People have moved from whole genomes to whole exomes to really targeted sequencing to really answer the region of interest. So what we have today is Jim Hartigan presenting how to accomplish your NGS goals with RainDance Targeted Sequencing. So Jim Hartigan is the East Coast Field Application Scientist at RainDance Technologies, and he really gets involved in pre-sale product demonstrations as well as post-sale installation, training, and on-site support. He's held a number of managerial roles and really extensive experience in both the first-gen and second-gen DNA sequencing space. So it's a tremendous asset across all applications. He holds a BS in biology from the University of Massachusetts Amherst. And just a reminder, a video recording of this webinar will be posted on our website, and you can access that right after the webinar. With that, I turn it over to Jim Hartigan.
Hello, everyone. Thanks for attending. I saw the registration list, and there are quite a few familiar names as well as some new names to me. So what we like to do today is talk about the technology behind RainDance and how that can help you achieve your goals. So today's agenda, first we're going to go over the advantages of what it is that we do, the science behind what we do if you're not familiar with that, and then move on to a little bit more of the background details. So how the primer design pipeline works, go over our automated equipment, the ThunderStorm instruments, as well as some aspects of the workflow that can save both monetarily and for your processing time. Then we'll move into, after everything that RainDance does, how that works into the benefits in the data sets. Okay, so let's get started. Okay, so the first topic is the massively parallel singleplex PCR. So what I'll go into is exactly what that is. When you're doing targeted sequencing or target selection, there are a list of items that are important for most of our users. That could be design coverage. Obviously, you want your region of interest to be covered at the highest level possible. Also, your sequencing coverage uniformity. How uniform your depth of coverage is after you sequence your selected target. The sensitivity of the assay, so how frequent of a variant or how infrequent of a variant you're able to detect with that particular target selection. The specificity, so how effective that target selection is. When you do your enrichment or your target selection, what percentage of what you ultimately sequence is then going to be your target relative to anything off target. Of course, a streamlined workflow is important, ease of use for a particular lab, making sure that the upstream and downstream applications are consistent with what your lab is already using is important to most folks. Automation is very important, not only for the streamlined workflow aspect, but also for reproducibility. So you want to eliminate as many of the manual steps as possible. Many of our customers also want random access testing. And what that means is if there are multiple assays in process in a lab at a given time, that each one of those can be accessed very easily on a given day, depending on what the lab needs to do. And of course, everybody wants it to be cost effective. And we'll show later on in the presentation how RainDance helps you achieve that. So the RainDance solution really comprises three different parts. The first part is your gene panel, so the genomic content that you're interested in. Of course, that's an important component. With our gene panel, so you can target up to 20,000 different locations within the genome. And typically what we get is a 99% region of interest coverage. So whatever region you happen to be interested in, ultimately you're going to select roughly around 99% of that. Secondly is the automated instrumentation that we have. That's our ThunderStorm system that can process up to 96 samples. And then the consumables where the target selection actually happens. The consumables we refer to as chips. The chips are the microfluidics where the target selection actually happens. Those are manufactured by Sony, and they use the same technology that Sony uses to produce Blu-ray discs. So it's a very reliable process. So because of the scale of Sony and because they're experts in this particular field, it's also quite economical. The chips can process 2 samples per chip, and we'll get into that a little bit later on in the presentation as well. And ultimately, what this all wraps up to is right around a 3 times sequencing cost savings. So everything that we do on instruments allows for a much more efficient target selection that ultimately saves you money downstream in your sequencing costs. Okay, so how it works. What RainDance does is massively parallel singleplex PCR. And the way that we do that is by doing the singleplex PCR in droplets. On the instrument, picoliter-sized droplets of both the pre-made primer panel and your sample of interest are merged. That individual droplet contains an individual PCR primer pair, so you ultimately get a singleplex PCR reaction. Now that's done a millionfold, so it's millions of individual singleplex PCR reactions. And I'm stressing the singleplex PCR because we'll get into how that benefits a little bit later in the presentation. So you can think of these individual droplets perhaps as individual test tubes or individual wells within a plate. It's just that all of those individual PCR reactions happen in an individual tube at a very, very small scale, so a picoliter scale. After that amplification, your pool of targeted product is then ready for sequencing on any of the next-gen sequencing platforms. So it's not specific to an individual platform, nor is it tailored to an individual platform necessarily. So the first aspect is the primer design pipeline. So in order to target your region of interest, you of course need to design towards that target of interest. So our primer design pipeline, again, it's a traditional singleplex PCR. So we're designing 2 PCR primers per region of interest or per amplicon. The primer design pipeline uses primer 3 as a backbone with proprietary additions. And the input can be any number of things. If you have gene IDs or RefSeq IDs, if you have a contiguous region, it can be genomic coordinates, it can be gene symbols, and all of this is transferred to RainDance via a simple Excel spreadsheet. One of the nice things about the input is that when you're doing your primer design, that you'll have an assigned field application specialist to work with you on the upfront experimental design. So if you have questions along the way, there's somebody there to guide you at every step, as well as to lend guidance of what we've seen work best in the field. And we can also tailor the primer design based on instrumentation that you use in your lab. So read lengths, for example, might be important on how you're designing the primers. Or it could be a factor of the input material. So you'd want to design primers for FFPE samples a bit differently than if you had high molecular weight genomic material. And you have somebody there every step along the way to assist you with that. The design output, what you receive back once we do the primer design, is, again, an Excel spreadsheet that details all of the primer information along with the amplicon information. What we typically see is a 99% success rate, so a 99% region of interest coverage. That 1% could be highly GC-rich regions, it could be homologous regions, but we typically don't see too much of an issue designing even through those areas. And one thing that we also can do is take the 1% that wasn't successfully designed on the first pass and put that back through for a second pass. What I normally see in the field as far as a finalized design is a 100% region of interest coverage. So along with the summary information, again, you also receive information on the primer sequences, amplicon sequences, bed file information, things that are very easily converted into genome browsers or for any internal review that you might need to do. So after we've done the in silico primer design and that it's finalized on your end, the primers are then synthesized. We use one of the larger primer vendors, a very well-known primer vendor. And we put those individual primer pairs into droplets at RainDance. Those primer droplets are then delivered to you as a gene panel or a library vial. Those individual droplets, again, they contain individual primer pairs, but all of your primer pairs. So a given vial or a given gene panel contains primer pairs designed to amplify your entire region of interest. Those droplets are going to be completely evenly represented, so each individual amplicon has the same number of droplets contained within any panel. So, of course, if you receive the primer vial, you need to actually do the merge. So we have instrumentation that we've designed to do that. That's our ThunderStorm system. ThunderStorm system can process anywhere between 2 and 96 samples in a given run. There are 8 panels, the random access that we've described earlier. If your lab happens to have multiple assays that are in process at the same time, in a given set of up to 96 samples, you can access up to 8 different assays. The vials that we deliver are either 8 use or 48 use, meaning that the vials have enough droplets, let's say, to process either 8 samples or 48 samples. And that really just depends on the throughput in your lab. And they're chilled on instrument. So on instrument, those vials are going to be stable for 30 days. Off instrument, at 4 degrees, they're stable for a period of a year. There is a on-instrument barcode scanner. So if your lab has an upfront LIMS system, or if you use barcodes to track things, it's capable of doing that, and of course also to log the reagents that are used on instrument. The throughput is 48 samples a day. So like I said earlier, each chip processes 2 samples at a time, and each chip takes roughly 1 hour to process. So after the sample comes off of the instrument and you have an amplified pool of your target of interest, you still need to take that amplified pool and get it onto your sequencer to do the sequence detection. Most people will currently be using a sequencing library construction approach. So what we've tried to do is completely eliminate the need for sequencing library construction. And we've done that with what we call DirectSeq. The DirectSeq application follows the standard workflow for the on-instrument portion. So you prepare your samples either with sheared high molecular weight genomic material or from FFPE, depending on your experiment. It's mixed with PCR cocktail. The PCR cocktail is very standard with TAC, mag chloride, nothing that is very out of the ordinary. You enrich the samples on instrument. We've gone over how the instrument works. The primary PCR will amplify your target genes. After that, there's a secondary PCR that builds on the sequencing adapter and any index that you might want to use for barcoding purposes in your sequencing run for multiplexing. The way that that works is the primary PCR utilizes a universal tail, the red portion in step 3. That universal tail is the same for each one of the individual PCR primer combinations. The universal tail is actually the 3' most end of your sequencing adapter. So when you put that primary amplified pool into a secondary PCR, you can then build on the remainder of your sequencing adapter as well as the index. That's shown as C and D in step 4. What this allows is you to build on the sequencing adapters using that secondary PCR, which is a quick 30-minute PCR cycle, rather than going through the full sequencing library construction, which is not only time-consuming but oftentimes quite manual. So essentially, what this allows is the RainDance workflow to eliminate a lot of the steps in the middle and get from the point of DNA extraction to sequencing with quite a few less manual manipulations and just fewer steps overall. So there are, of course, other requirements to the process. So one of the most important features is the design coverage that we've already talked about. Our customers typically want 100% of the region of interest covered. We're able to achieve that, and typically what I see is a 99% region of interest coverage on the first design attempt. The other aspect is coverage uniformity. The reason why that's important is because you want each one of your amplicons in the pool, or each one of your targets in the pool, to then be equally represented when you do your sequencing. If there is a large disparity between amplicons or between targets, what you end up having to do is over-sequence some of the amplicons relative to others. Having uniformity allows you to best utilize your sequencing capacity. The sensitivity and specificity are important. Sensitivity meaning how low of a minor-most allele you can detect. If you have a heterogeneous sample, say a tumor sample that has normal mixed in, you might have only about 1% of your variant present in the cell population. So it's important to be able to detect that low. The specificity is related to how efficient the enrichment process is. So how much of what you have enriched shows up in your sequencing population. How much is on target. Again, the random access testing, very important to people that have multiple panels active at the same time. And then, of course, it needs to be cost effective. So the design coverage, again, that we typically see is greater than 99%. This chart shows a number of different panels. The rightmost y-axis is the target region size. We can design large regions of interest or small regions of interest as long as it fits within the 20,000 primer pair maximum. The left axis is the design coverage that we were able to achieve, which is represented with the green line at the top that you can see regardless of the panel size, hovers right around 100%. We're also able to provide better target coverage, and what I mean by that is being able to target repetitive elements, pseudogenes, homologous regions, high GC content regions, where some other techniques aren't necessarily able to do so. Many of those repetitive elements are intronic, so might not be of interest to some people. But as you can see here, some of the repetitive elements are also exonic. So exon number 1, which is shown here, is typically relatively high GC and difficult to capture if you're using a technique that only has an individual probe. When you have 2 PCR primers, you're then twice as likely to be able to find at least one of those that is unique enough to be able to confidently capture your region of interest. You can certainly capture repetitive elements if you don't have a PCR-based approach, but because they're repetitive, you're also going to be capturing the off-target region, and so your data ultimately is going to be questioned as to where it's correctly mapped. The other aspect is uniformity. It is shown here. If you go across left to right, this is the relative sequencing coverage of those regions of interest, and you can see that it is fairly uniform all the way across the region. That's also shown here in a study that was done at UCSD. The paper is available for download on our website. And what this was a comparative of different PCR-based approach. RainDance, which again is a singleplex TCR, versus some of the multiplex options that are available. And what you can see in the chart in gray on the y-axis is the percent of amplicons that are within a given fold of the mean. Okay. What this is a representation of the normal distribution that you're going to get from your sequencing coverage. And what it shows is that a high proportion of the amplicons in our capture were very close to the mean coverage. So it's a representation of how broad your normal distribution is. And this is important because, again, anything that is not robust, you're going to have to sequence more in order to get it to an appropriate coverage level for you to call variance. If you have amplicons within the pool that are very robust, then you're going to have to over-sequence those. So you're using sequencing capacity to over-sequence amplicons if your normal distribution is very broad. Okay. The other requirement is the sensitivity. So how low of a minor allele can you confidently detect? How low of a minor allele frequency can you confidently detect with the assay? We've shown through multiple tests that we can detect as low as 1% in a heterogeneous sample. This is a study that was, again, performed at UCSD using our off-the-shelf cancer hotspot panel. It's a panel that is designed for 54 cancer-causative genes with greater than 13,000 mutations from the COSMIC database. So the next note is the random access testing that we've talked about a couple of times. Again, on instrument, you can load up to 96 different samples. Of those 96 different samples, you can access up to 8 different panels. The 8 different panels are loaded on the instrument and can process either 8 or 48 samples, depending on how you chose to order. And those are able to be accessed independently and in any configuration in those 96 samples that are loaded. So, for instance, if you had one test that was very low throughput, you might only have 2 or 4 samples, while another you might have 40 samples in a given day. Any configuration that you would like is perfectly acceptable. So what all of this wraps up to is a significant savings on the downstream sequencing. So with a high coverage level, you're able to reduce any Sanger fill-in that might be needed otherwise. The specificity that I talked about, the on-target component, how effective that enrichment is, it really helps you utilize your sequencing capacity, so you're not sequencing things that you're not interested in. The uniformity really plays quite a large role in that if you have uniform concentrations of amplicons in your enriched pool, you're then able to multiplex to the highest level without having to over-sequence some amplicons to bring others up to the same relative coverage levels. We've seen this equate to roughly a 3 times sequencing savings, simply by being able to multiplex more samples together. The other factor is the direct-seq application that I described, which eliminates any need for the sequencing library construction, not only saving in the reagent cost there, but also saving time and lab personnel time as well. So this is a slide that we touched on at the beginning, and that we've talked about all of these bullet points throughout the presentation. And the point here is that we're able to meet really all of the requirements that most of our customers are looking for. So the high design coverage, the high region of interest coverage, roughly 99%, 100% in most cases, the coverage uniformity, the cost savings by being able to multiplex, the sensitivity level, where we're able to go to as low as 1% minor allele frequency, depending on the experimental needs. The specificity, where we're able to achieve 90 to 95% on-target reads with our direct-seq approach. The streamlined workflow, so eliminating a lot of the hands-on effort that it would take to enrich via other techniques, with the automation in conjunction with that. The random access testing, where if your particular lab happens to have a few different assays that are all in process at the same time, that on a given day, any of those can be accessed independently of others. And the fact that it's cost effective. So the savings that I've described with the 3x reduction in sequencing needs relative to other approaches, as well as the cost savings with direct-seq, eliminating the sequencing library construction portion. So over the last few years, as the company's grown, we've seen a pretty widespread adoption of the process and different utilization depending on different institutions. So the map here is perhaps missing a few red dots. As we place instruments on a daily or weekly basis, you're going to have more and more, so it's a little bit difficult to keep up with. But I think that the point is that we're a global organization, and also that we are placed in a number of different types of institutions. So anywhere from the large genome centers to the larger cancer centers, medical genetics, and service providers as well. And of course, this isn't an extensive list of our customers. It's just a representative showing. And of course, as our customers adopt our technology, they are publishing quite rapidly. This is just a few select publications from some of the different institutions that we work with. Okay, so this is a summary of what we've talked about. We're going to come back to this, and I think that right now we'll probably open the floor to any questions that you might have. We'll probably need just a moment to look through some of the questions that have come in, and then we'll come right back. So we have a few questions that have come through relative to the primer design and using either custom primers or primers that have already been designed and that you're comfortable with in your individual lab. We have no problem at all building those primers into a gene panel. Of course, we can't speak to the efficacy of those primers, but if you would like to use something that you've already designed, that's perfectly acceptable. Okay, there are also a few questions that came in about the number of genes that can be within a panel. As long as the number of genes fit within the 20,000 amplicon maximum, that's fine. Of course, genes are going to be different sizes. What I typically see is an average of about 50 amplicons per gene, and most of our panels that people ask to be designed end up between 1,500 and around 4,000 amplicons. So that's the size that most people use this application for. Okay, there's a question about how many samples that I can use for my library. That is going to be dependent on the library size. So it depends on the number of primers that we're able to then convert into droplets. As a generality, with a panel of the size of 1,500 to 4,000 amplicons, like I was talking about, you're going to be looking at roughly 3,000 samples. But that depends a lot on the ordering pattern and expiry times, things like that. The next question is relative to the nature of most of the panels that we design, whether they're custom applications or whether they're for whole-exome purposes. They are for custom applications. The exome is too large to fit within the 20,000 amplicons limit that I was describing, and really the technique is best suited for custom applications on the order of 50 to 150 genes, somewhere in that 1,500 to 4,000 amplicon range that I was talking about. We can certainly go lower or higher than that, depending on the experimental need, but that's typically where we see customers end up. The next question is about the confidence level that we have that an individual droplet contains a PCR primer pair that will correctly amplify the region of interest. The way that we're confident in doing that is the primer design that we do goes through an in silico amplification to predict whether it's going to amplify only the region that we're interested in or perhaps an off-target region. If the primer design pipeline detects that there's also an off-target region, that is going to go through a secondary design process to try to restrict the primers to only the unique region of interest. There are also quite a few questions coming through about the sequencing platforms that we're able to work with. The technology can work with any of the next-gen sequencing platforms. Essentially what you're going to get from your enrichment is a pool of PCR products. Those PCR products are the same as what you would get from a traditional Singleplex PCR. It's just that they are performed in picoliter-sized droplets in an emulsion. The next question is on the input requirements for FFPE samples. That input requirement is currently 250 nanograms. We're working to reduce that, but haven't rolled that out yet. 250 nanograms of FFPE is our lower limit. The next question is on the amplicon sizes. So during the primer design process, again, you have an application scientist that will work with you. One of the questions that we'll be asking is what sequencing platform you intend to use. Based on the read length of your particular platform, we can design the amplicons either smaller or larger. The other determining factor is the type of input material. So with FFPE that's typically degraded, we would want to design the amplicon smaller, whereas with high molecular weight, we would want to design a bit larger. Just as a generality, with FFPE samples, we would design to a 200 base pair maximum, and with high molecular weight material, we would design to a 600 base pair maximum. Okay, we have two different questions on the turnaround time. So there are two components to the turnaround time. One is the primer design process. From the time that something is submitted to your application scientist, the turnaround time to get the in silico design complete is typically a week or less, depending on the size of the region of interest. From the point of design approval to delivery of the gene panel or vial is 6 to 8 weeks for a new panel. If the panel had been synthesized in the past, it might be a bit shorter than that, but 6 to 8 weeks is typically where we are.
Hi, this is Ram, and there are quite a few questions coming in about pricing as well as access. So I'll address a couple things. One is on the website, there's a service provider page where you can access the list of service providers. So you can actually find out what panels are being offered for your region of interest, and you can contact us through raindancetech.com and fill out the form, and we'll be in touch. There's also quite a bit of pricing questions. We have your name and question, and we'll pass it through to our sales rep, and they'll be in touch with you to give you your kind of custom pricing.
Okay, it looks like that's all we have for time for the question portion. If anybody has the need for more information, you can go to raindancetech.com/contact, complete the form, and one of our representatives will be in contact with you to answer any questions you might have. Thank you all for coming. I hope everyone found it informative. Have a good day.